Good afternoon and welcome to this webinar, High-Throughput Assessment of Compound-Induced Cardiotoxicity using Human iPSC-Derived Cardiomyocytes, brought to you by Xconomy and presented by Eurofins. My name is Howard Choi with Xconomy. Before we begin, let me explain how you can participate in today's presentation. First of all, if you have any technical difficulties during today's session, please press the Help button on your player console to receive assistance in solving common issues. We welcome your questions during today's event. In order to submit your questions to today's presenter, simply type your question into the question window on the left-hand side of your screen and then hit the Submit button. We will be answering as many questions as possible during the Q&A session that will follow the presentation, but feel free to send in your questions at any time and we'll add them to the queue. If at any time you are having audio difficulties or the advancing of the slides, simply hit F5 to refresh your webcast console. And please be aware that today's session is being recorded and will be made available on the Xconomy website, xconomy.com, beginning tomorrow for your review. So you will be notified by email when the archive is available. And now, I'd love to introduce today's presenter, Panida Lertkiatmongkol, PhD. She is a senior scientist and group leader of cell-based screening at Eurofins DiscoverX. Panida, the floor is yours.
Thank you for the kind introduction. Good morning, everyone. My name is Panida Lertkiatmongkol. I'm a senior scientist and a group leader of cell-based screening team from Eurofins Discovery at San Diego, California, the United States. I will be presenting an application of human iPSC-derived cardiomyocytes to assess cardiotoxicity of compounds. This paradigm is engaged by comprehensive in vitro Proarrhythmia Assay, or CiPA, for cardiac safety during drug development. Our agenda for today will include background on cardiac and ion channel, how ERG is important for cardiac safety, and how it becomes a limitation in drug development. Then, we will learn about comprehensive in vitro proarrhythmia, or CiPA, which is a new paradigm for cardiac safety evaluation, and how Eurofins Discovery takes part in CiPA initiative. For our new service on cardiotoxicity, I will discuss about our calcium flux assay service using human iPSC-derived cardiomyocytes that I will show the validation data. Then, we will conclude the talk with the application of cardiomyocytes in cardiotoxicity assessment and Eurofins Discovery CiPA assay services. First of all, let's get to know our hearts. The heart is a pump that is driven by electricity. In a minute, it can beat from 60 to 80 times. That means that it's about 100,000 times in a day, or 35 million times a year, or 3 billion times in a lifetime. Therefore, if beating of our heart is interrupted, just only a few minutes, even if that's so, this is so detrimental as multiple organs can be damaged. It may even cost us a life. We can look at activity of the heart by measuring electrical activity of the heartbeat. This starts with an electrical impulse that travels from the upper chambers of the heart to the lower chambers. The electrical impulse stimulates the heart muscles to pump blood from the upper heart. The electrical impulses are initiated by the upper chambers that generate the first wave, called P wave. After that, the bottom chambers generate the next wave, namely QRS complex. The final T wave represents the electrical recovery, where the bottom chambers return to the resting state. Consequently, if these waves are distorted, function of the heart can be abnormal. Ion channels expressed in the heart are the key to the healthy waves. The heart expresses several ion channels. ERG, a human ether agogo-related gene, is a potassium channel that relays relaxation signal or repolarization to the heart muscles. ERG is promiscuous, as it can bind to diverse kinds of compounds. Inhibition of ERG can prolong the QT interval. However, this is not detrimental unless early after depolarization or EAD occurs. Opening of calcium channel or late sodium current during the prolonged phase can result in EAD, leading to Torsades de Pointes or TDP. Therefore, ERG inhibition can lead to QT prolongation, but only together with EAD can lead to TDP. Drug-induced QT interval prolongation and Torsades de Pointes arrhythmias are the leading causes for compound attrition during drug development. In 1998, tafenidine or ciladine, which is a non-sedating antihistamine that can induce QT prolongation and arrhythmia. Sertidol is an antipsychotic drug that also induces QT prolongation. Then, in 1999, estamazole or mismanol is a non-sedating antihistamine was found to induce QT prolongation and arrhythmia. Gefloxacin is an antibacterial that can induce QT prolongation. In 2000, CiPA or Torsades, which is a gastroprokinetic agent, was found to induce QT prolongation and arrhythmia. Consequently, in 2000, S7B was introduced as a guideline for non-clinical testing strategy to assess the potential of compounds to delay ventricular repolarization. These example drugs are potent blockers and highly selective in the therapeutic ranges, since the events of TDP are far too low to be detected in clinical trials. QT prolongation is used as a surrogate marker. Then, how can we relate IC50 of ERG to cardiac safety in clinical trials? This is a quote that poison is in everything, and nothing is without poison. The dosage makes it either a poison or a remedy. We use safety margin, which is calculated from RC50 divided by free plasma concentration. For ERG, if the safety margin of a compound is over 100, then the compound is preferred for further development. But if the ratio is below 30, the compound shouldn't be continued for further development. Eurofins Discovery offers an assay to estimate free plasma concentration of compounds together with ERG inhibition assay for evaluation of safety margin. Since blocking ERG non-QT prolongation does not necessarily mean TDP, Comprehensive In Vitro Proarrhythmia Assay, or CiPA, is an initiative to facilitate adoption of a new paradigm to assess clinical potential of TDP. The new CiPA paradigm is comprised of mechanistically based in vitro assays coupled to in silico reconstructions of cellular cardiac electrophysiologic activity, then verified completeness through comparison of predicted and observed responses in human-derived cardiomyocytes. To further verify clinical potential TDP of compounds, human-induced pluripotent stem cell-derived cardiomyocytes are used to mimic effects of the compounds against heart muscles. To obtain iPSC-derived cardiomyocytes, skin samples are collected and grown in cultures. Then proteins are introduced into the skin cells, and these cells differentiate into cardiomyocytes. iPSC-derived cardiomyocytes can be used as a model to evaluate potential cardiotoxicity of compounds. Using cardiomyocytes as a model, we can perform calcium flux assay to monitor contraction and relaxation of cardiomyocytes. We detect either increased or decreased levels of calcium ions using calcium-sensitive dye. The dye binds calcium ions in cytoplasm as displayed on the left panel. The dye elicits fluorescent signals that can be captured by a high-speed CCD camera in FLIPR, which is a real-time imaging platform. Shown on the left panel is the picture of iPSC-derived cardiomyocytes from Fujifilm CDI. I grow these cells in a 384 well plate for the calcium flux assays and use the new FLIPR Penta, which is equipped with a high-speed camera that can capture 30 frames per second, compared to the previous Tetra that can go up to 6 frames per second. This allows us to obtain high-resolution data as displayed on the left. The top panel is the vehicle control, and the lower panel is the xylitol-treated cells. You can see that xylitol, which is a high-risk TDP compound, causes prolongation of the calcium oscillations compared to the vehicle control. To define calcium oscillation traces, we choose 3 descriptors, which are mean peak rate that demonstrates how many primary peaks observed in a minute. Peak spacing is the space from the beginning until the end of the peak, and calcium transient duration 90 or CTD 90. Moreover, we can observe the secondary calcium spike that is an EAD-like waveform. The detected fluorescent signals from the calcium flux assay represent intracellular calcium oscillations. In order to understand what happens to cardiomyocytes upon compound treatment, I used PeakPro2 displayed on the top left panel. PeakPro2 is a model of ScreenWorks software of FLIPR that we can use for extracting kinetic parameters from the calcium traces. PeakPro2 can assign primary peaks, which are identified by blue dots. It can also set the baseline and the triggered levels, as shown in red and green lines. The increase in fluorescent signals corresponds to contraction, while the decrease corresponds to relaxation of cardiomyocytes. We can monitor in real time and over time. In this example, the second panel on the left is the pre-treatment trace. Then, the cells were treated with compounds and incubated for 30 minutes and read on FLIPR, as shown in the third panel on the left. There are several parameters that we can extract from the traces using PeakPro2. From our experience, mean peak rate, CTD 90, and peak spacing are pretty consistent across the whole 384 well plate and from experiment to experiment. So, we can go ahead with these parameters to plot our dose response curves for calculating RC50s, as shown in the right column. There are more examples of cardiomyocytes treated with isobutyrenol, which is an agonist of beta-adrenergic receptor that can increase the heart rate. The trace exhibits increased mean peak rate and decreased CTD 90 and peak spacing. The next one is astamazole, a non-sedating antihistamine that is categorized as intermediate risk of TDP. Estamazole is a potent ERG blocker and causes QT prolongation. From the trace, the mean peak rate is decreased, while CTD90 and peak spacing are increased, which are characteristics of peak prolongation due to blockage of repolarization by ERG. The last one is xylitol, which is categorized as high risk for TDP. Xylitol is a beta blocker. It can also prolong repolarization by blocking the rapid component of delayed rectifier potassium current. From our assay, xylitol-treated cells exhibit decreased mean peak rate, but increased CTD90 and peak spacing, showing peak prolongation as well. For these three parameters, the data points are quite tight and clearly distinguishable between pre-treatment, shown in round, and post-treatment, shown in navy. These scatter plots demonstrate that our calcium flux assays is uniform and consistent. The observed effects are also correlated with actions of the compounds. Using our assay, we can monitor in real-time cardiotoxicity of E4031 at different time points from low to high concentration. At low concentration of 8 nanomolar on the top row, we observe peak prolongation and secondary calcium spikes at 30 minutes post-treatment, while at very high concentration, such as 500 nanomolar, at 30 minutes post-treatment, we observe cardiac arrest. The effect of 500 nanomolar is very fast that we can observe peak prolongation as early as 20 seconds post-treatment before the cells undergoing cardiac arrest. Again, this is to reiterate that we can observe either stimulating or blocking effects of compounds compared to the cells treated with vehicle control. Moreover, the secondary calcium spikes, which are EAD-like waveforms, are observed in the cells treated with xylitol, which is a high-risk TDP compound. We can demonstrate that treatment with xylitol results in peak prolongation and secondary calcium spikes correlating with its high-risk TDP. We can also plot dose response curves and fit the curves of these three parameters to obtain EC50s or IC50s. As you can see that across different parameters, the resulting EC50 or IC50 are pretty much in the same range. In summary from my talk today, we learned that the heart can pump blood normally as long as the beating is not distorted. However, irregular beating can be detrimental, especially in the events of TDP. Therefore, drug-induced QT prolongation and TDP should be considered to reduce the chance of compound attrition during drug development. In order to get away with this, so that is the CiPA initiative, which is a new paradigm for TDP risk assessment. For us, Eurofins Discovery, we provide services to evaluate cardiotoxicity of compounds as engaged by CiPA initiative. In this webinar today, we show that using human iPSC-derived cardiomyocytes, we can assess cardiotoxicity of compounds through our calcium flux assay. The resulting phenotypic data of the assays are validated and can be helpful to mitigate TDP risk during drug development. Thank you for attending our webinar today. If you have any more questions or if you would like to learn more about our services, please go to the website shown on this slide and you can contact me through my email. If you have any questions, we are welcome to answer. Thank you.
Thank you, Panida. A few of you have already submitted questions, and while our presenter is answering your questions, please take a moment to check out the resource links for additional information and for the link for the upcoming Eurofins webinar. Panida, I'd like to ask our first question from the audience. How does cardiotoxicity assessment contribute to drug discovery pipeline?
Thank you for that question. So basically, as you have seen from my presentation, that in the beginning, when you would when you would want to submit your compound for further drug development, or when you have a drug candidate that you would like to submit to FDA, but first of all, you would have to make sure that your drug or your compound would be safe enough not to be harmful to humans, and especially like it wouldn't target the ERK ion channel, which is kind of something that you don't want it to happen, in a way that it would block the channel. And once it blocks the channel, then the adverse effect can happen. So basically, that is so critical to make sure first that your drug is safe, that it would not cause arrhythmia. And then later, usually after people would do start with primary screening or secondary screen, and then once you have the drug candidate, so then you would have to make sure that your drug is effective and then it's safe enough to further go into the in vivo study or in clinical trials. So this assay is very useful in a way like before you would actually test your compounds in vitro study for safety. So basically, you can use our model to first of all see whether it would have the effect on the heart beating, and basically people can do the assay on ion channels, specifically like ERG or KV channel or else. But the useful of cardiomyocytes is that you can test, you can do phenotypic screenings where you don't have to think about which target to screen against, but then you use the cardiomyocytes, and then you can see like the effect that it would go to any channels. That basically this is a good model instead of having to test against individual ion channels targets. Yeah.
Thank you, Panida. Sweet. Yes, Sarah, go on.
No, that's it.
Okay, thank you.
Question.
So we have a few questions coming in, and for those of you viewing, feel free to continue to submit your questions as the discussion continues, as Panida will be on hand for the next few moments to answer your questions. We have another question from the audience, Panida, for you. Do we have a sense of what false positive or false negative rates are regarding in vitro cardiotoxicity assays and clinical concordance? Or how sure can we be in these assays regarding risk reduction?
Okay, that's a very good question. So basically, what we have done in the past is that before we would start to provide a service, so then basically for ourselves, then we have done some assay development, and we want to make sure that our assay would be helpful and beneficial for your drug discovery pipeline. So basically, we would have to validate our assays that if we have the compounds that these are the control compounds that we use in our study, and for example, like estamazole or xylitol that I showed in my slides. So those compounds, it's been known since in the past that these compounds, they have cardiotoxicity effect. So basically, we started with those compounds where you already know the clinical data is being well characterized, and then we perform our assays to see whether we would see any correlation to the clinical data. So then after that, then we can see whether if some of the drugs should have, it should have peak prolongation, it should have Torsades de Pointes, and do we see that in our assays? And from our assay development, So it's very robust, and in a way that we could see the clinical effects that could be related to our data as well, in a way like in my slide. So then you could see that, for example, esomeprazole and xylitol, these two drugs, they are known to cause arrhythmia, right? And then for esomeprazole, it's categorized in CiPA panel as the intermediate risk for Torsades de Pointes, whereas for xylitol is categorized as high risk Torsades de Pointes. So we could see peak prolongations in these two compounds, as well as the secondary calcium spike, which is a kind of similar to early after depolarization waveform. So basically, if you would see these two markers together in the phenotypic screenings, so that can imply that your compound may have similar effect to the reference controls compounds. So that's where we can, relate to the compounds or the drugs that they have been characterized well with in vitro data and in vivo data or clinical data. So that's how this is helpful in a way, like we validate our assays with the compounds that have been known for its effect already. So then we can use these compounds as the reference for you to further, test your compounds whether it would have similar effect to the reference control compounds. And basically, for cardiomyocytes, so about the false positive or false negative rates. So this is one of our concern as well, that we may have, false negative effect that we might not expect it to be, or that kind of thing. So then basically this is where we would want to make sure that when we perform the assay, so we can reproduce the data. And for our control compounds, then we have done independent assays several times in the past already to show that the way that we optimize the assays, it would not cause any artificial effect. And then we have the controls in the plates where we screened. So then to make sure that our assays is reproducible, and we also have duplicates in the same plate or on different independent day to see if the data is consistent across each experiment that we have performed. So some of the compound that if it should have, for example, isoproterenol, that it should increase the heart rate, because this is also the agonist of adrenergic receptor. Our assay is good in a way that you don't have to target only ion channels, but you can also screen the effect, that from GPCR receptor as well. So this is another control that we tested, and we could see that it's increasing the beating rate, and this is consistent, but we have not seen it to, have the opposite effect. So this is where we could verify whether the compound would increase or decrease the heart rate. So we have these controls in there. And our cells, we take care of the cell very well, and then we make sure that they are even and uniform when we perform the assays. So then, and then we have at least 3 replicates of that same concentration, and so then it should reproduce. But sometimes in the past, I could see some effect in a way that we have 3 wells of the same compound, same concentration, but sometimes 2 wells, they show the same phenotypic data, whereas the other well, the cell would be, silent or kind of cardiac arrest. So that kind of thing can happen too. So basically, that's where when you do this kind of assays, this is more finicky than just other cell-based assays, that we would prefer to make sure that at least we have 3 replicates for the assay, to make sure that this is what we see, and it's also another interesting thing about this effect is that it can be the result from genetics as well. So when we use the cardiomyocyte, again, these cells, they are human-derived stem cells. So each time it's not the same clone. Then it's also is a pool of cells from different donors. So each time that can be different effects or slightly different. But one good thing about it is that if it would have, for example, peak prolongation, so then you will see that. But from time to time, it can be like how much it would prolong the peak. So that can be different. But if it would show that effect, so yes, it would show that effect regardless of if you would test it from this cell vial or other vial. So the response can be different in a way, like for how far it would go. But if it would have the effect, then it would show the effect. Yeah. So then, in terms of our assay validation, we have done pretty much in the past year before we would launch this service. Yes.
Thank you, Panida. And can you speak a little bit about what kind of compounds should be tested for cardiotoxicity?
That's a good question. So about this assay, so again, even though it says cardiotoxicity assessment, basically you can also screen the compounds just similar to other cell-based assay if you want to screen against some targets as the target of your compound as well, besides testing for cardiotoxicity. So first of all, you would get the cardiotoxicity profile for sure from the compound if you already have the compound, and then you want to make sure that the compound would not have cardiotoxicity effect. So that is the first benefit of this assay. And the next one is if you want to see whether the heart beating pattern would be something that you want to target. So then these assays can also be used as the screening targets as well if you want to perform, if you want to like see whether your compounds can modulate their heart beating. So it's in addition to see if it would have cardiotoxicity effect, but if it's something that you want it to do, right? So then you can also use this assay as well.
Thank you, Panida. We have a question from the audience as well, that just came in. Are the data collected at physiological temperature?
That's a really good point. So exactly, this is where the assay has been maintained at 37 degrees the whole time. When it's really the whole time that, for example, if I would have to see the cells, then it's in the safety cabinet hood. So that's not 37, but I do that really quick, and media was wrong when it was doing that. So then after that, I put the plates right in 37 degrees right away, and during the time that we performed the assay, so everything is kept warm, like the compound plate or our instrument. We also keep it at 37 because these cells, they are very sensitive to temperature shift. So this can affect the activity of the cells. And another important thing is that the temperature is important for the activity of the cells, for consistency of the data, and if there is a shift, so then the data would not, it would be kind of like noisy, and then it wouldn't be consistent. So for this assay, it's more finicky in a way that I have to really make sure that every single step that I'm doing is really at 37 degrees. So then it will not affect the activity of the cells, to maximize the activity, to make sure that it's beating, because if the temperature slow down, so that can affect beating rate, that it would go slower.
Thank you, Panida. And as a follow-up question from the same attendee who just asked the previous question, as a service, do you also do voltage-sensitive dyes besides calcium that you have shown here?
Okay, so that's another thing that we would want to target in the future. So thank you for bringing that up. Basically, right now, we only offer the calcium dye assays, but the voltage-sensitive dye, we have not, we are not able to provide this service yet. So in parallel with the calcium flux assay that we discussed here today, even though we don't have the voltage sensitive dye assay yet, not yet, but then I guess you can also look at our MEA assays, which is kind of, you also measure action potential of the cells. So this is where we also have the other assays in parallel that for iPS cell-derived cardiomyocytes. Right now we have two options. One is to do calcium-sensitive dye, and the other is to do MEA assays or patch clamp. That's a good question. So that will be in the future that we will keep, growing our portfolio in cardiotoxicity. So it would be coming in the future. So we just have to, take time to make sure to do assay development, then that we can bring out, the best data, the best service to the customers. So then it will take some time. And like for cardiomyocyte assay calcium flux, so this one, it took me like almost 2 years before we would launch it. So just to make sure that we can do it as best as we can before we would provide the service. So that is also another good thing that for the voltage sensitive dye that it would come in the future. So stay tuned.
We will certainly stay tuned, Panida. And another question that just came in from one of our viewers. Are you using a chemically defined medium?
Chemically defined medium. So basically, cell media is, we usually like is the media that, the components that in the media. So it's like that, but some of the components could be valuable, for example, like FBS. So that can't really be defined exactly as is kind of like the serum, but yes, we do. To make sure that each time when we grow the cells, so then they get the same treatment. Yeah. For so then we can reproduce the data. That's a good question. Thank you.
Great. Just a reminder, you can submit questions using the questions box on the left and just click submit, and Panida will be on hand to answer them. Our next question that we have on the list is, can the assay represent clinical data?
Yeah, so again, thank you for this question, and this is where I can relate back to the previous questions that I got earlier. So these assays, again, we use our control compounds that these compounds, they were already well characterized and in vitro data or in vivo or clinical data. So we use those to as our reference when we perform our assays, and then we can see that we can relate that back to the clinical data and like kind of to relate the phenotype. So then yes, we already validate our control compounds, and then we can use these information as reference for the test compounds that we will screen in the future. So it's basically something like you can have as reference, but again, that can help you to see whether it would have similar effect to other reference compounds. So it's not really a solid answer whether this would be something that is exactly like the reference compound, but you have the idea of how it would show the effect based on the reference compounds data. So then further, you can have the idea of what you would pursue in the future for your test compounds and how you would do clinical, how you would, whether like you would want to pursue in clinical trial or in vivo data, in vivo study.
Great, thank you, Panida. And we have another question that just came in from a viewer. Do you have insights, or could you provide insights into genomic profiles to help stratify patients by potential risk profiles, i.e., if you see variability in assays due to genomics, is there enough of a pattern or correlation between genomic profiles and drug responses?
That's really a good question, and that is actually something that we have in our mind, that we are aware of genomic variability. So when we started to plan our assays, so then it's basically we use only for specific population, like from female, and the age range of the donor is from 30 to like 40 something. But it's also something like the people are different, and each person would have different response, right? So then basically that's really a good question to, that the genetic effect and drug response. This is something where you would have to keep in mind that when you test the compounds, so would have to make sure that you also test in, different populations, not just only to specific population, because we are all different. So right now, what we would pursue in the future, we would offer to test different genetic populations in the assay. So basically, you can also see the effect of your test compound in different genetic populations. So then that can be helpful because, like, for when you do clinical trials, you want to make sure that you do not pick only specific population, because when you want to commercialize the drugs, it's going to go to anybody on this world. You don't want to sell it just only to specific population. So this is also one thing that we would like to pursue in the future, that we will offer different genetic profiles for cardiomyocytes. Then you can see the effect of the drug in different genetic populations. Yeah, so because we don't know whether like this drug might not have much effect on peak prolongation or EAD-like waveform, but it might have in other populations too. So then this is where this will be something in the future that we will offer too. Yeah, but this is a good question. Thank you.
Great. And another question that just came in. Are all of your reference compounds synthetic? And do you have any natural product scaffolds?
Oh, this is another thing that, thank you for the question and bringing this up. So what we tested and verified, so it's been synthetic compounds or synthetic drugs, but I have not tested like natural products yet. So that's also something that we can try to test those compounds in the future too. Yeah, we begin with the compounds or the drugs as our reference controls, and we pick the compounds where they are already well characterized for in vitro data, in vivo data, and clinical data. So then we can correlate our assays to these data, and then if it's something like not well characterized, so we would not start with those compounds yet. But once we are confident that, okay, our assay is validated and it can correlate to the clinical data. So then we can, continue to work on other compounds that we don't know a lot about it, because we already know how to interpret our data and how our data can relate to the clinical data or the in vivo data. So then we can use this information for other compounds that they are not well characterized. But sorry, no, we have not tested natural product compounds yet, no.
Thank you, Panida. There are no further questions from the audience. So with that in mind, I'd like to thank Panida and the Eurofins team for an outstanding discussion. And thank you to our viewers, of course. Please take a moment to answer the feedback form that appears at the bottom of your screen. And actually, Panida, we have a question that just came in from another attendee. So does rhesus negative blood type have an effect on vaccine/drug use? O negative, A negative, and B negative. The last two are rarest.
Yeah, so there are some studies on genetic variations and then drug response. So the RH type, like plus or minus, or blood group A, B also, all these is basically the variations of the sugar modification on protein. And basically that is an indicator or a marker of, because people can have different glycosylations, and this is where people are different, even though they express exactly the same protein, but the glycosylation modification can be different. So this is where this is another indicator of ion channels. So the function of ion channels, for example, like we have nicotinic receptor or the NMDR. So those, they need some glycosylations on the ion channels, and basically it's not a direct implication of this blood type would have effect on the drug response or the efficacy or the use of the vaccine, but it's because if you would likely have this kind of glycosylation where you can detect easily through your blood group, so that can imply how your receptors or ion channels are glycosylated as well. And glycosylation can affect activity of ion channels or the receptor. So this is not really like a direct implication, but it's kind of indirect on, you can imply like what, like this blood group may have this kind of glycosylation and the other would have different, and that also affects the ion channels activity. So yes, it can affect the response if you would do the assay. So the drug may react differently because glycosylations can modify the activity of the ion channels and the receptor. So yeah, but we have not tried, like for cardiomyocytes, we have not looked specifically yet on the different blood group of the donors. So this is where I have the answer of how it would be different yet, as we have tested only from one kind of the donor. So that will be in the future where we would offer different genetic pools for cardiomyocyte to be tested. So that would be interesting to look at whether like people from different genetic populations, and then how the drug would have response in these different genetic pools. Yeah.
Great. And another question just coming in right now. Which platform do you use for MEA measurements?
Oh, it's Hamamatsu. So we have this in Essentials. This is also under Eurofins Discovery.
Great, thank you. Thank you, Panida. There are no further questions coming in. So with that in mind, again, I'd like to thank you, Panida, and the Eurofins team for an outstanding discussion. And for you viewers, please take a moment to help us improve our webinar delivery platform by answering the feedback form that appears at the bottom of the screen. And on behalf of Xconomy, have a productive remainder of the day. Thank you.